It looks like a single creature, pulsing in the darkness. Yet every inch is made from thousands of bodies, all born from one egg, fused into a living monster that can stretch beyond the length of a blue whale. This is not science fiction. It is the reality of siphonophores, colonial predators whose very existence challenges what it means to be an individual. But where does the colony end? And where does the monster begin? Siphonophores belong to the phylum Cnidaria, a group that also includes jellyfish, corals, and sea anemones. Unlike their solitary relatives, siphonophores exist as colonies, each one an intricate assembly of specialized bodies, all genetically identical, joined together in a single living chain. The best-known member of this group is the Portuguese man-o'-war, often mistaken for a jellyfish, but actually a surface-dwelling siphonophore that drifts on ocean currents with a gas-filled float. Deep below the waves, other siphonophores take on forms that are far more extreme. Some species stretch over 40 meters in length, rivaling or even surpassing the size of a blue whale. These giants remain almost invisible in the open ocean, their glassy bodies so fragile that even the gentlest current can tear them apart. For decades, their true scale and complexity went unnoticed because traditional nets destroyed them on contact. Only with the rise of remotely operated vehicles have scientists begun to witness siphonophores fully intact, drifting through the darkness like living ropes. The sheer length and delicacy of these colonies defy easy comparison. They are not just long, they are composed of thousands of individual units, each performing a specific role, all moving and feeding as one. This arrangement puts siphonophores at the far edge of what it means to be an animal, not a single creature, yet not a loose collection of individuals either. In the vast taxonomy of ocean life, they stand as a reminder that nature's categories are rarely as simple as they seem. Every siphonophore begins as a single drifting egg, no different from the millions of other planktonic cells scattered across the open sea. Fertilization triggers a transformation. That one cell divides, then divides again, building the foundation for something far more complex than a solitary animal. Soon, a tiny larva forms, known as the protozoid. This is the founding member, the original body from which the entire colony will arise. But instead of growing into a larger single organism, the protozoid sets up specialized regions called growth zones along its slender axis. Within the growth zones, a process unfolds that is both methodical and astonishing. New bodies called zooids bud off in sequence. Each zooid is a perfect genetic clone of the original, produced without sexual recombination. The term zooid refers to these multicellular units, each one technically an individual animal by ancestry, yet never capable of living on its own. They remain attached, linked by tissue and a shared internal canal. As the colony elongates, the growth zones continue their work, budding off new zooids in a precise species-specific order. This production line can run for years, adding hundreds or even thousands of new members to the chain. The youngest zooids cluster near the growth zones, while older, fully developed ones are carried further down the stem. The result is a living assembly, stretched across meters or even tens of meters, all originating from a single egg. Every zooid shares the same DNA, but soon each will take on a specialized role, a living demonstration of how one becomes many and how many become one. Along the length of a siphonophore, specialization is built into the very architecture of the colony. Each segment, or cormidium, is a repeating module composed of distinct zooid types, arranged in a precise order along the central stem. 5 main classes of zooids form the backbone of this living assembly line: nectophores for propulsion, gastrozoids for feeding, gonozoids for reproduction, dactylozoids for defense, and palpons with roles in digestion and excretion. Every zooid is a multicellular unit shaped by its function, some with muscular bells for swimming, others with tentacles lined with stinging cells, and others dedicated to the production of eggs or sperm. This modular design is not random. Each chormidium contains a set pattern of zooids, and the sequence repeats dozens or even hundreds of times down the colony's length. In some species, a chormidium might include a single feeding polyp, a pair of defensive zooids, a palpon, and a reproductive unit, all linked together. The arrangement is species-specific, the result of tightly regulated developmental signals as new zooids bud from the growth zone. What binds these thousands of units into a single organism is the unifying internal canal system. Every zooid connects to a continuous gastrovascular cavity, a hollow, fluid-filled tube that runs the entire length of the colony. When a feeding zooid digests prey, nutrients flow through this shared cavity, reaching even the most distant swimming bells and reproductive structures. Waste products and chemical signals travel the same route. This internal circulation acts like a bloodstream, distributing resources and information, and making the colony far more than the sum of its parts. A siphonophore's predatory reach begins with its tentacles, long trailing filaments that can stretch for meters into the water column. These tentacles form a nearly invisible curtain, drifting and undulating in the deep, ready to intercept anything that wanders too close. Along their length, thousands of specialized stinging cells, nematocysts, stand primed for action. Each nematocyst is a microscopic capsule loaded with a tightly coiled harpoon tipped with venom. The instant a small fish, crustacean, or plankton brushes against the tentacle, the nematocyst fires. The discharge is almost instantaneous, measured in microseconds. The harpoon pierces the prey's outer layer, injecting toxins that paralyze or kill in moments. Multiple nematocysts can fire in rapid succession, overwhelming even the most agile target. Once the prey is immobilized, the tentacle contracts, drawing it toward the nearest feeding zooid. There, digestive enzymes break down tissues, and the resulting nutrients enter the colony's shared internal canal. This system ensures that energy from a single captured animal is distributed across the entire organism, feeding swimming bells, reproductive units, and defensive zooids alike. In the open ocean, where food can be scarce, this method maximizes every opportunity. Dozens or even hundreds of tentacles can operate at once, each working independently but contributing to the colony's collective survival. The siphonophore's hunting strategy is not just about catching prey, it is about turning the entire colony into a living, coordinated net. A predator that feeds as one, no matter how many bodies it contains. No single brain steers a siphonophore. Instead, a diffuse nerve net stretches through the colony, linking thousands of zooids into a network of sensation and action. Along this living cable, nerve impulses travel at speeds of 75 to 150 centimeters per second, fast enough to synchronize movement across a body that can span dozens of meters. When swimming bells contract, they do so in a coordinated wave, each pulse perfectly timed with its neighbors. This rhythm is not set by a central command, but by electrical and chemical signals that ripple through the nerve net, allowing each zooid to sense and respond to the activity around it. The result is a colony that glides as one, shifting direction or speed with a fluidity that rivals any single animal. Siphonophores achieve unity not through hierarchy, but through a web of distributed control, where every part listens and answers in real time. Growth in a siphonophore never truly stops. As long as the colony survives, new zooids bud from growth zones near the front, extending the living chain ever further into the darkness. This process is relentless, yet the entire structure remains astonishingly delicate, about 95% water, with tissues so transparent and soft that even a gentle current can shred them. Traditional nets destroy these colonies on contact, leaving only fragments for study. Only with remotely operated vehicles have researchers observed siphonophores fully intact, drifting through the deep sea. Attempts to keep them alive in captivity have failed. The smallest disturbance or shift in water conditions is enough to unravel the colony. This extreme fragility, paired with continuous growth, blurs the boundary between individual and collective. Where does one animal end and another begin when the body is both endless and ephemeral? In the deep ocean, individuality dissolves into something stranger, a living network, thriving where few can follow. As we probe farther into Earth's last frontiers, siphonophores remind us, nature's greatest monsters are built from unity, not division. The boundaries of self are still being rewritten, far below the surface.